Good morning. Today we're going to discuss how life began. I think it's a common question that lots of people have, and scientists have worked on this question for nearly over 100 years, thinking about how life began on Earth. To understand the theories about how life began on Earth, you need to understand cells. Just a little bit about cells. We'll go into cells a little bit more in the next unit, but cells are the basic unit of life. It's the simplest thing that I can take from you and keep alive on its own. It carries out those 7 properties of life we talked about in our first lecture of the semester. Now, all cells on Earth are then divided into 2 groups, prokaryotic cells and eukaryotic cells. So I want to explain the differences between these particular cell types. So if we start with prokaryotic cells, these are cells that do not have a true nucleus. That means that the DNA is loose in the liquid part of the cell. There is no membrane containing that particular nuclear material, the DNA. And there are also no other membrane-bound organelles inside of a prokaryotic cell. So there's no membrane compartments to do different reactions. In the fossil record, we see prokaryotes arriving in our fossils in colonized mats at about 3.5 billion years ago. Because this group shows up in the record, the fossil record first, and because of the chemical structures of the cell and the lack of the true nucleus and these compartments, we think that this cell type evolved first. This cell type includes bacteria and archaean, those 2 big domains of life. Remember, there's only 3 of them, so 2 of them on planet Earth are prokaryotic. The second kind of cell type is a eukaryotic cell, and this is a type of cell with a true nucleus. This means that you can see over here our DNA, or our nuclear material, is bound inside of a lipid container, or a membrane is covering it and houses it separately from the liquid and the other organelles of the cell. Eukaryotes also have, besides the nucleus, they also have lots of other membrane-bound organelles. Like over here, we have the rough and smooth ER, the mitochondria, we have a Golgi apparatus. So these kinds of vacuoles, we have these kinds of organelles that also are in their own membrane, separate containers, right, in the liquid of the cell. We see the first fossilized eukaryotic cells in our fossil record at about 2 billion years ago. And so this is why we believe, and also like sort of the membrane-bound sort of compartments, we believe that the chemical structure and the fossil record tell us that this cell type arrived second on planet Earth. This includes our 3rd domain of life, domain eukarya. This is probably the domain that you know. It includes protists, plants, fungus, and animals. All right, so scientists at the turn of the last century especially were sitting down almost in think tanks and thinking about how do we go from non-living material to building prokaryotes, right? So prokaryotes arriving in the fossil record, in the cell structure, we think that those came first on planet Earth. And so how do we go from first non-living materials to these prokaryotes? And then the second stage was how do we go from the prokaryotes to the eukaryotic cells that we see on planet Earth. So when scientists were thinking, they came up with a series of steps. These were the basic things that needed to happen in order to build a prokaryotic cell from non-living material. And I'm going to present it with 4 different steps that are necessary to go from non-living material to these first cells on Earth. So the first step is taking non-living material and building simple, almost like Lego bricks that are necessary and required for life. It just so happens that living things are built from 4 macromolecules. Those macromolecules include proteins, carbohydrates, lipids, and nucleic acids. All 4 of these macromolecules then have a simple, single, repeating structure. So proteins, for example, their simple monomer or repeating structure is called an amino acid. A carbohydrate has a single unit sugar, like glucose, that repeats over and over again. lipids have a single unit called triglycerides, or like a triglyceride that repeats over and over. And nucleic acids have something called a nucleotide that repeats over and over. So the first step of our hypothesis is going from non-living materials to these single unit monomers, like amino acids, simple sugars, simple fats like triglycerides, and these nucleotides that build our genetic information. The second step of our hypothesis, we need to take those single units and then make them form polymers, or more complicated repeating units of those single units. So think of a pearl necklace and think that you're like adding beads to this necklace to give it complexity. A single unit might not be able to form like sheets and helices and chains and complex structures. So step number 2 is taking those single units, those monomers, and building polymers, those more complicated sheets, maybe circular structures, those more complicated chains, sheets, helices that we see in our macromolecules. Step number 3 of our hypothesis, we need to be able to form genetic information that can be passed from parents to offspring. And so this is the formation of our fourth kind of macromolecule, our nucleic acids, which include DNA and RNA. Those are built from the repeating units called nucleotides. And so we need to be able to build that last macromolecule so that we can basically inherit instructions from parents to offspring about how to build these cells and then keep these cell lines going. And step number 4 in our hypothesis is we need a container. We need to be able to put these compartments and these different chemicals and macromolecules and keep them separated from the external world. And all cells on Earth have a lipid sort of outside surface, a membrane we call it, and this lipid surface actually keeps the internal liquid contents of the cell separate from the external liquid world. So we need to be able to form that lipid membrane surface for building a cell. Now, if we can go through these 4 steps, then we can get from non-living materials to our first prokaryotic cells on planet Earth. So how did scientists actually go through these steps? So at first, at the turn of the century, scientists were almost like thinking through it as like a theory. We need these things in order to build our first cells from non-living material. But then in 1953, scientists started to actually test each of these steps to see if we could actually do it in a laboratory setting. So they took step number 1, the famous experiment was a Urey-Miller experiment, and they took step number 1 and basically broke it down and thought to themselves, can we put all the non-living sort of minerals and water and things like that were in early Earth, can we put that in an apparatus and actually build these single unit monomers for our 4 macromolecules of life? So Urey-Miller built an apparatus like this where they included an ocean, they included an atmosphere with water vapor, they added heat to it to simulate lightning and sunlight and condensation in that particular process. They added different gases in the atmosphere that might have been popular or common in early Earth, like methane, some nitrogen-based gases, H2 hydrogen gas, and then they allowed this to cycle. They allowed it, the ocean, the pretend ocean or simulated ocean, to be heated up. It would evaporate into the sort of atmosphere, mix with these gases, and then condense back as rain. And then periodically, the researchers would actually collect samples, almost these sludges, and within even a few days, this apparatus went from kind of clear gases and liquids to kind of this brown sludge. And they started testing for different things. One of the first things that they tested for were amino acids. And within even a few days, they could get some very simple amino acids being formed in these particular sludges. So they were able to show in a simulated, sort of an early Earth simulation, that we could go from non-living materials to these simple single units that were necessary to build our 4 macromolecules. Urey-Miller did this experiment in 1953, but other experiments then tweaked the gases of the atmosphere, the liquids in the ocean, and they were able to get not only amino acids, but also simple sugars, simple lipids, ATP, which is it's a kind of nucleic acid that builds energy for all of our cells, and some simple like genetic material as well. So they were able to sort of show in an early Earth simulation, going through that step number 1, going from non-living materials to building these single units. Step number 2, they did something similar where they had these early Earth simulations and they would drip the contents over like sort of hot rocks and things like that, simulating what early Earth might have looked like. And researchers looked to see if they could take those single units and then form more complicated chains, sheets, and other sort of polymer structures. So going from those amino acids to building different kinds of protein, more complex protein structures, or going from those single unit sugars to more complicated carbohydrates. And so they were able to do this as well. Step number 3, could they create simple nucleic acids, simple genetic material that would enable us to store information and pass it from like parents to offspring so that we could continue cell lines? DNA is actually complicated. It contains a double helix. It almost looks like this in these letter L shape. These are nucleotides, and then they repeat in this like sort of ladder structure. RNA is actually single-stranded, so it's a little bit more simple in terms of like it's a little bit smaller, typically shorter, and RNA can really do everything that we need it to do inside of our cells. Because RNA has this simplicity, because of the kinds of nucleic acids that Urey Miller, those later experiments, what they were like kind of looking at, researchers hypothesized that probably RNA was the first kind of nucleic acid to form. And then DNA, which is more complicated with its double nucleotide structure and this ladder, this helices structure versus a single strand, probably came later. It's a little bit more stable. When we get a nucleus, we can house it and protect it inside of that space. So it kind of makes sense in terms of like sort of the chemistry, the complexity, and what we saw in these early Earth simulations, that RNA probably came first and then DNA. But we were able to create sort of these simple predecessors to this nucleic acid or these genetic materials that are needed in order to pass information from one generation to the next. And lastly, could we create a container? Scientists actually could put liquids and lipids, oils and fats together that are common for this membrane boundary. And even by shaking them just a little, they were able to get liposomes. These were kind of early, simple structures, right, with a lipid boundary. And you create these when you do dishes, right? When you do dishes and things like that, you can see bubbles and things forming in the liquid, separating sort of oil pockets from the liquid environment. These pre-cells with this lipid boundary then could be formed in the lab. So scientists worked through these 4 steps and they looked at whether they could simulate these 4 steps. We haven't necessarily gone from non-living material in a lab all the way to creating a new cell line or something like this, but they definitely have like thought through each of the steps and what it would require to go from non-living material to these first cells, and then also simulated parts and pieces of those steps going forward. This 4-step hypothesis then presents our theory, right, on how we go from non-living material to get to those first prokaryotes on planet Earth. Now, the second part of how we get cells on Earth, or how we get life, is going from prokaryotes to eukaryotes. Remember, prokaryotes show up in the fossil record at about 3.5 billion years ago, and then eukaryotes at about 2 billion years ago. So we've got a little bit of time, and eukaryotes are known for their true nuclei with this membrane-bound container, and also all kinds of compartments, these organelles that occur. And we think that eukaryotes evolved from a 2-step process. The first step is kind of the outside membrane in these liposomes can pinch and fold, and in cells, even vesicles, we call them, are little pinched off pieces of membrane that travel throughout the cell. These kinds of pinching off and folding of the outer membrane, we believe, would have been step one to create the endomembrane system of organelles. The endomembrane system of organelles, even today, is a system of organelles that are connected through this lipid boundary, either by directly touching another lipid boundary or by these little packets or these vesicles that transport between one part of the cell and the other. The endomembrane system would have formed not only in this first diagram the nucleus, but also the folding off of the rough and smooth ER, the vacuoles for storage, the Golgi apparatus, all the way out to the plasma membrane. All of those membranes are continuous either by direct contact or by these indirect vesicles transporting between different parts of the cell. There are some cells that are outsiders. These outsiders have their own loops of DNA. They have their own sort of way to divide themselves. They are not connected into this sort of membrane bound system, or this endomembrane system of organelles. And this includes, over here, we're showing the mitochondria in orange and a chloroplast in green. These are independent organelles, again with their own loops of DNA, and they can divide themselves. And we think that those independent organelles arose from prokaryotes engulfing other prokaryotes. The two pieces living as symbionts, right, in a mutualistic relationship over time until the inner sort of prokaryote loses its independence. So if a prokaryote engulfed another like kind of heterotrophic prokaryote, we could sort of see forming a mitochondria. If it engulfed a photosynthetic prokaryote, then we could see losing independence and gaining sort of a chloroplast over time. Those loops of DNA have been researched both in the mitochondria and the chloroplast, and we can actually research and link them back to different kinds of ancestors. Like we think the chloroplast might have come from something like a cyanobacteria, one of these photosynthetic prokaryotes that still occur on Earth's surface today. So that is the second step. Endomembrane system organelles occur by folding and pinching off of the membrane. And then the second step is creating these sort of engulfing other prokaryotes and creating these independent organelles, or the mitochondria and chloroplasts, for examples. This then leads us to our 3 domains of life that we see on planet Earth. Here is the root, so time beginning with non-living material, time where our first cellular life began, and then we have bacteria, archaean, and finally those eukaryotes sort of developing. So this is all I want to say about sort of microbe evolution. Next time we'll look at the whole tree of life and some of the differences of living things on planet Earth. So see you then.